So the topic is about the prediction of protein binding sites. So basically this is a project I have done in last 2 years. So what is the binding site? The binding site I'm going to talk about in this study is the binding site between proteins and the large macromolecules such as DNA, RNA, and proteins. So if this is an example, so here is the PDB ID. So basically these are the protein sequence and this is the DNA chain. And if we take a look at this area, if we zoom in, we'll find the sum reduced in the protein is very close to the DNA chain. Then we can define this reduced as the binding sites on the protein. So basically in this study, I will use 6 angstrom as the distance threshold for the definition of the binding sites. So of course, there are already some binding sites predictor existing already. For example, Summon A from Ross Lab, which can predict the DNA and RNA binding sites. But the problem is all these methods are training based on only the radio level information, which makes them can only predict which makes them cannot predict whether the input protein is a binding protein or not, or non-binding protein. So in most of the cases, the users were not familiar with their input protein. So if they input a non-binding protein, or this method will still report some positive prediction, which obviously will affect the performance. So to solve these issues, we designed a new prediction approach. So basically it's a 2-level prediction. So in the first level it's a protein level. So we first decide whether the input protein is a binding protein or not. So check whether it is a the protein binding protein, a DNA binding protein, or RNA binding protein. So if it gets a positive prediction in the first level, then we will move to the second level, the residue prediction, to decide whether which reduced on the protein is actually the binding sites. So with such approach, so even the users do not know whether their input protein is a binding protein or not, we still can have a relatively good performance. And also, if the users are very familiar with their input protein, they can choose to only run the residue level prediction. So here's the dataset I used, including the protein DNA interface database, protein RNA interface database, and the protein from Ross Lab. And I also use geoannotation data in protein level training. So as geoannotation do not have the residue level information, but I can still use it to create the negative data in the protein level training, as well as collect the non-binding proteins. And I also use it to enlarge the positive data in the protein level. And after I train the model, I will need to do the comparison between my method with the others. So then I create an independent dataset based on the PDB sequence. Here's some pre-processing for the dataset before I train the model. So first I map the DNA sequence onto uniprot sequence with shifts. And then I remove the redundancy with h-value 0, and I also need to keep there no redundancy between my training set and the independent set, because I don't want to overestimate the performance of my method. And then I use 5 for cross-validation for the training set. So basically I split the training set into 5 parts. So every time I train the model on the red one, the training part, and optimize the hyperparameters in the cross-training part, the green one, and finally I evaluate the performance on the blue one, the test part, and I do it in 5 iterations. So now we can move to the first step, the protein level training. So in protein level training, I tested 2 different algorithms. So first is profile kernel with supported vector machine, and the second one is Word2Vec. So profile kernel is a sequence alignment based method, which means it depends on the cyberlast. And Word2Vec is a natural language processing based method, which means it is alignment independent, and the alignment independency will make the Word2Vec much faster than the profile kernel. First one is profile kernel. So to talk about profile kernel, I need to say something about supported vector machine first. So the mission of supported vector machine is to find a hyperplane with a maximum margin, which can have the best performance to separate two groups of samples. And but sometimes you cannot easily find a hyperplane in the input feature space. Then you need to use a kernel function. A kernel function can map the samples into higher dimension. And with a correct kernel function, then in the higher dimension, you can find a hyperplane. And profile kernel is exactly a kernel function. So here is an example for the profile kernel. So first, you use the Cyblast to do the sequence alignment, then you get the profile. So here each value represents the frequency of 20 amino acids at each position in the sequence alignment. And there's a parameter in k-mer called in profile kernel called k-mer. So here is an example for 3-mer. So 3-mer means instead of consider one amino acid, now you consider frequency of a combination of 3 amino acids. So then you check whether the frequency of 3 amino acids passed a threshold or not. So basically, with 3-mer, you will map the protein sequence into 8,000 dimension. So 8,000 because for 3 amino acids, you will have 20 times 20 times 20 equal to 8,000 combination. So basically, it's 8,000 dimension. The second method I tested is O2vec. So O2vec is a natural language processing algorithm which can represent the word in vector. And I used the skip-gram neural network model. So there are two different models for O2vec. First is called Sabor. So Sabor means you use the neighbor word to predict the central word. Where skip-gram means you use the central word to predict the its neighbors. So here is an example show you how O2vec works. So for example, you have a sentence like this. Then you apply a window approach to scan through the sentence to collect the pair of samples. So basically, the pair of samples is the central word and its neighbors. So after you get those samples, you can train a neural network. Basically, it means you use the left side, the central word, to predict the right side, its neighbors. So here is the neural network. So first, you have the input vector for the input word. It could be one hot encoder vector. Then let's assume you have a hidden layer with 300 neurons. So for the output layer, it's a softmax classifier, so which means each value here represents the probability of each word in the vocabulary to be the neighbor word of the input one. After you train a neural network, you will have the hidden layer weight matrix, and you take this out to use it to create a word vector. So basically, you have the input vector, and you times it to the hidden layer weight matrix. Then you will have the word vector. So basically here the length of the word vector will equal to the number of neurons in the hidden layer. So in my study, I will consider 3 amino acids as a word. So after you get the word vector for each word, to represent the whole protein sequence, you need to sum up all the vector for all the possible 3 amino acids in the protein sequence. So you will have the vector for the whole protein. Then you can test a different classifier algorithm based on this vector. Then this is a comparison between two algorithms. So basically here each value is the F1 score. And here the red one is profile kernel, represent the profile kernel. And the blue is ProtoVec or O2vec, the different name but same thing. So we can see here in most of the tasks, the profile kernel outperform the ProtoVec or O2vec, but in protein binding prediction, so ProtoVec is slightly higher than profile kernel. And I also tested other natural language based method like ARMO, but profile kernel still better than others. And another thing we noticed is as profile kernel is a sequence alignment based method, so profile kernel is more good at to predict the protein from large family size. And as O2vec or ProtoVec is alignment independent method, so it is more good at to predict the protein from small families. And I also including the homology-based inference in my method because in our lab previous study we found about the subcellular localization prediction. The combination of machine learning and homology-based inference achieve a relatively good performance. So the homology-based inference means that, for example, you have protein A with the experimental annotation, but the input protein B do not have. Then if protein A have a similar sequence with protein B, then we can transform the annotation from A to B. And this is how I did it. So first you have the orange pie, the proteins, all the proteins with experimental annotation, for example, the DNA binding. Then I take a non-redundancy part from it, which means all the protein sequences in this part do not have similar sequence with each other. Then I blast the input protein against this part to find whether it can hit some similar sequence. So if it can hit some similar sequence, then we can transform the annotation to the input protein. And this we can see here in the comparison, the machine learning plus homology-based inference achieved the highest score. Here the black bar. And now we can move to the per-residue level prediction. So in per-residue level prediction, I used the typical neural network. It has the input layer and a hidden layer and an output layer with two nodes. So one for binding and one for non-binding. And the features I used including the PSS metrics, the profile, and like the secondary structure prediction or chemical properties. So normally the final prediction will driven on based on the binding node minus non-binding node. So if binding minus non-binding give you a positive value, it will be a binding prediction. If it gives you a negative value, it should be a non-binding prediction. But but in some situation like this one, so in this example, the x-axis is the protein sequence, and each bar represents each position, and the y-axis is the final raw score. Means if it's larger than 0, it's a binding prediction. If smaller than 0, should be a non-binding prediction. But according to the biological knowledge, we know that the binding sites are usually clustered together and shown in groups. So for this prediction, it is okay. They have a higher score and they in groups. But for this one, the reliability score is close to 0 and it's isolated. So according to the biological knowledge, we may supposed to think this one is false positive. And in this situation, this one, according to the raw score, it should be a non-binding position. But again, the reliability score is close to 0. And it is in the middle of a bunch of very high reliable prediction. So we may think this one should be also be a binding site. So that this one may be a false negative. So to solve these two issues, we used a secondary filter. So now we do not use simply the binding minus no binding. Now the final score is the average score for all the positive in a window approach. So which means instead of consider the only one amino acid, now we also consider its neighbors. So here the performance of the secondary filter. So here the red bar means prediction with the secondary filter, and the green one means without filter. So we can see the secondary filter significantly improved the Q2 accuracy compared to without the filter in all three tasks. And finally, I do the comparison with other method based on the independent dataset. So here, the method with the smear phase is my method. You can see in all three tasks, it achieved the highest performance. So this comparison is the protein level prediction. So we predict whether the protein is a binding protein or not. So here we can see some methods achieve the same performance because all these are residue level prediction. And when I apply them on protein level prediction, they just simply try to predict all the protein as the binding protein, which makes them achieve the same performance because the coverage will be 100% and the precision will be just the fraction of binding protein in the whole data set. And they are such high just because in my independent data set, the binding protein is the majority. And this is the comparison in the residue level. we can see again my method achieve a highest score. Only except in RNA binding prediction, there's one method achieved a similar performance with us. And for the reliability, you can use the method from GitHub, or you can run Predictor protein server, and you can achieve the performance of the method. Thank you very much.
Okay, so that's the second half on protein ligand binding prediction. I'm Maria, and I'm going to cover basically everything that JJ didn't cover. So all other binding molecules, so small binding molecules. First of all, something that you probably all already know, but just as a quick recap, a bit of the biological background. So why are we interested in predicting protein ligand binding? Of course, it is an important aspect of a protein's function. If a protein doesn't bind something, then it probably doesn't function correctly. And if you know the binding site of a protein, it gives you insight in different aspects of the functionality. So you first of all know or can know what reaction is catalyzed if it is an enzyme. You can also know how the functionality is regulated. So if you have certain allostatic binding sites, you can identify if there's a certain molecule down or upregulating the functionality. You can also know where the binding site is located, which also can help you in further investigating the function of a protein. And you can also identify which mutations might disrupt the binding site of a protein. So if you know which mutations are disrupting in a protein, and you also know where the binding site is located, you can just easily map that, and then you know what mutations might destroy the binding site. The next thing is, so when we are talking about binding, what can a protein bind? So the first few things first two bullet points, but the first few ligands we already heard about. So that's proteins, DNA, and RNA. These are macromolecules. And everything else is basically a small molecule. So we have metal ligands like magnesium or calcium or iron or whatever. We have peptides, which are proteins up to 30 amino acids usually. And they are different from proteins because it's not a protein-protein interaction. So they don't form a complex and they don't function together, but it's actually just a protein binding a certain peptide and then doing something with it, degrading it or whatever. It can also be an antibody. So things like that are peptides, basically. And then the most common thing that normally binds a protein, if it's not a macromolecule, is a small ligand. So that's basically everything else that is not covered with the other things. So ATP, ADP, single amino acids, whatever comes to mind. All of the molecules that are not covered by the other 4 bullet points. The thing now is, if you want to predict binding residues, we usually want to go for a machine learning approach, and then we need a labeled data set, so we need to know binding residue annotations for our proteins. And that's mainly the difficult part about binding residue prediction, because getting these annotations is not that easy as for other cases. So the first point to go to is, as always, PDB. You usually need a structure to identify the binding site, because so for example, for localization, you can make an experiment where you have a fluorescent image and you see, okay, a certain protein is located in the nucleus without knowing its structure, but you cannot identify the binding site without knowing the overall structure, at least not experimentally. So what people do is they do a co-crystallization experiments where you have a protein structure bound to a ligand, and you just crystallize that complex. And then you can identify the binding residues just by defining everything that is close to the ligand as the binding site. And that's already one problem, because how do you define close to the ligand? So usually you have cutoffs like 4 or 6 angstroms depending on what binds, because it also depends on the size of the ligand. But you could also argue that only the free catalytic residues involved in a reaction are involved. Yeah.
But doesn't it make more sense to look for the residues that are essential for it? Like when you change other residues and the binding still occurs, then they shouldn't be relevant or they shouldn't be part of the binding site.
You could also do that, but that's even more complicated to do as an experiment, because then you need to have a knockout experiment. And the problem is that you have many things that you can knock out that will destroy the binding site, even though they never interact with the ligand. So that's basically the other side of the coin. So if you want to define the binding site, there are many residues involved in forming the binding pocket, but the question is, how long do you still define them as being part of the binding site? Because you have a lot of residues that stabilize the binding pocket as a structure, but that never actually interact with the ligand. And you could argue for example, for an enzyme, you have this catalytic triad, you have 3 residues that actually are interacting with the ligand and catalyzing the reaction that is occurring. But of course, if you mutate everything around it, it will also not work anymore. So that's always a bit of a trade-off, like what is actually involved in the binding site. But that's actually the problem. So two other things that are, or one big thing that is caused by two things that is also a problem for binding annotations is basically that not all ligands that are present in PDB are biologically relevant. So what does that mean? So first of all, what can happen is that you have a ligand bound in the PDB structure that is not actually similar to what would bind to the protein in vivo, or that you have small molecules that are just part of the crystallization experiment that never actually bind to the protein in nature, but that just happen during the experiment, so they're like just experimental artifacts. So how can we deal with these two things? So first of all, regarding the low similarity, we have to discuss one thing, and that's the bound ligand versus the cognate ligand. So the cognate ligand is what a protein actually binds in vivo. So what we would expect it to bind at least. And if the cognate ligand binds, so the cognate ligand is usually a concept for enzymes, so if the cognate ligand binds to an enzyme, then the reaction also occurs. I mean, that's the whole idea behind that. The ligand binds, the enzyme catalyzes a reaction, then you get a product out of it. But if that happens during a co-crystallization experiment, you don't get a protein-ligand complex because the reaction just happened and there's no ligand left. So what people do in these co-crystallization experiments, in fact, is they don't use the cognate ligand, but they use a ligand that is very similar to the cognate ligand. So it should bind in the same way, but the reaction should not occur. So for example, you could just replace ATP with GTP. So we would assume that it still binds in the same way, but the reaction cannot happen. But there we already have the problem that if we have something that is very, very similar, this approach can work and we can get something that is at least very close to the actual binding site. But you also have differing degrees of similarity. Maybe you don't have a ligand that is actually similar to your cognate ligand, or the similar ligands don't work because they also cause a reaction or something. And that's something that you have to take into account when you want to get a binding site annotation out of PDB. Because what Jiajun and Thornton did in this reference that I showed down there, they actually checked in PDB how often do we have something that is similar to the cognate ligand. So we have roughly 57,000 enzymatic structures in PDB. And the first thing that is interesting is that you have 17% of these structures don't bind anything, which is already surprising because it's an enzyme, it should bind something. So basically there we have a missing annotation. But of the remaining structures that are actually bound to a ligand, only 26% of these ligands have a similarity above 0.7. So that means everything below that you could argue is not really similar to what actually binds in nature. And if you have something, for example, that is much bigger, so you have a ligand that is much bigger than the cognate ligand, the binding site will also be much bigger. So you will annotate certain residues at binding that are not involved in binding in vivo. So you can filter for that, and that's basically what this paper does. So they provide a tool where you can, where they filter these annotations, and you can use these annotations because they're much more refined than what is in PDB. But it's limited to enzymes, so it's only a very small data set that you have this high quality set of residues, basically. The other issue that we have is that during crystallization, you need to add certain molecules and metals to just allow the crystallization to occur. And they are added in a very high saturation. So it can just happen by chance that a metal ion binds to the protein. That would never bind in nature, but just because of the high saturation, it can now bind to the structure during crystallization. And then you end up with a metal binding site in your structure, and you say, okay, we have a metal binding site here, but it would never occur in vivo. It's just something that occurred as an artifact during the crystallization. And you also need to filter for that somehow. And that's basically what BioLiP does. So BioLiP is a semi-manually curated database. And they try to filter out these irrelevant ligands. So to only focus on biologically relevant binding sites. So what they do, so the first 4 steps that are shown here are an automated process. So they start with a list of suspicious ligands, so ligands that are often used in crystallization experiments and that can therefore be artifacts. And then they take their new ligand and they check it against this list and say, okay, if the ligand is in that list and also it is repeated 15 times in the structure. So that means you have a PDB structure file, and for every binding site you have one entry basically. And for these artifacts you usually don't have 1 or 2 binding sites, but you have 20 or 25, just because it binds everywhere where it can bind. And that you can see, because then it is repeated 15 times and you know it doesn't make sense. Which protein should bind 15 times to, I don't know, any metal ion. Yep.
Why is the number so high at 15?
Because it is an automated process, just to ensure that you don't filter anything that might be actually biologically meaningful. So they just try to have here a very coarse-grained filter step, so something where they are really, really sure that it's irrelevant, and then they fine-grain it in the next steps, and then in the end they have the manual verification, and then they just throw out everything there that is still unclear. So it's just, the goal is to have a manual verified database, and everything that they do in the automation should be so coarse-grained that they can be sure that they didn't do anything wrong there. So that's why they chose 15. Okay, so if it passes that and you go to the next step, they basically look for contacts. So, I mean, if you have a binding site, you would expect that at least 2 or 3 residues are in contact with that ligand. It should not only be 1. And that's basically what they check here. So if you only have 1 contact, then it's probably not actually binding. And the other thing that can occur is that you have a small binding stretch, maybe 3 residues, but they are all next to each other, so it's residue 3, 4, 5. And then they also argue that it's probably not a binding site, because a binding site should be somehow a structural concept, so it should be a pocket or something should fold, and then you maybe have at least residue 3 and 7 or something interacting with the ligand. If it's only a short consecutive stretch, then it's probably not really a binding site, but just a metal ion attached to it that should not be there. And so they can again filter out some of the ligands as irrelevant. And then in the next step, they again check is the ligand that we are considering in this artifact list. If no, then they say, okay, it's probably relevant, and we will do a manual verification. If yes, they still don't discard it, because it can happen that you have a ligand that is usually used for crystallification experiments, which is in fact biologically relevant in this specific case. It happens rarely, but it happens. So what they do, they don't just discard it, but they actually check PubMed to see if they find any publication. Because if a certain small molecule is biologically relevant for this protein, we assume that there's a publication where it was at some point discussed that it is related to the function of this protein. So that's what they check for, and if they find a publication, then they also say, okay, it's relevant, and we move to manual verification. If they don't find anything, they just say, okay, it's irrelevant, and discard it. And then in the end, for everything that this automated process put out as relevant, they do a manual verification, they do literature research, check other databases, whatever, and try to identify if they're actually all relevant interactions. And then you end up with a database that is roughly 575,000 entries. So entries means one structure, one ligand binding site. So you roughly have 100,000 unique structures in there. So unique doesn't mean non-redundant, but so it can also be redundant structures, of course. And then you have 4 different kinds of ligands, so DNA, RNA, peptides, metal, and regular, so small. And you can see that small is by far the largest group with 265,000 entries right now. The thing is that these numbers are up to date, but only up to date for now. So BioLiP gets updated every week. So tomorrow the numbers will change. They will update it every Friday, so they will update it tomorrow again. That usually means that these numbers change by a few thousand. But it is a very redundant database. So in the second that you run a redundancy reduction on that, the number reduces a lot. And also the weekly updates don't change that much anymore. So it's not that you gain 1,000 proteins every week. It's just a bit refinement there, and a few structures added and a few structures checked, and so they're also working on this. So then just something passes manual verification and they just add it, but usually it's just redundant information. Okay, so that's BioLiP, and it's a very useful database because they actually deal with these kind of problems, and that PDB has these artifacts in there. Okay, so now we have a database, we can extract a dataset from there, and now we come to the interesting part: how do we actually predict these binding residues? And I mean, you have heard a lot about machine learning and predicting various aspects of protein function. So the question to you would be, do you have any ideas how you would deal with predicting binding residues? Also doesn't have to be machine learning, can also be something else. Yeah, Yannick?
Is the ligand given?
It helps if it is given, yeah, but I mean, in general, it's not given. I mean, you can predict what is binding, but usually you only have a protein, you don't know anything. But what would you do if you have the ligand? So what would it change?
Probably see if it has any special amino acids that encourage binding, for example, hydrophobic amino acids.
Yeah. Okay, so the first thing is what you can always do and what you can always decide is basically if you use something that is homology based or if you use something that is de novo, so basically template based versus template free. So for binding residues you usually don't do a full homology based inference, but you can do that, but you can also just look for binding motifs so that you know certain structural patterns that form binding sites, and then you just do a structural alignment and just see if they also occur in your structure, for example, or you do the same with the sequence. That also leads us to the next point. So that's also an important decision. Are you going to use structural information or are you going to use sequence information? And it comes back to your question, Yannick, if the ligand is given, the same goes for the structure. If you don't have the structure, we cannot use structural information. So usually structural information is much more precise in predicting the binding residues, but you don't have structural information for all proteins. So sequence information is probably worse in performance, but it just is broader applicable. And the same goes for template-based versus template-free, of course. Template-based works very good, it's really, really precise, but you don't have templates for everything. The next thing is the question if you should focus on a specific ligand type. So usually it helps a lot if you know what is binding, and it makes the prediction much better if you either distinguish at least small versus metal versus peptide versus DNA, whatever. But there are also predictors that actually focus on the actual ligand. So they just say, okay, I can predict a copper binding site or I can predict an iron binding site. And of course, again, these methods are much more precise than if you want to predict binding in general, because they can use much more specific information. But again, you have the problem, you need to know what your protein is binding. And for training, first of all, you limit your data set, because if you only predict copper binding sites, then yeah, you don't have these many data points. And also for predicting something, if I give you a protein, just say I want to know what is binding or where it is binding, you first need to identify if it's actually binding copper or not. And if it's not, then you don't have a predictor anymore. So that's not helpful at all then in the end. So that's always a question that you have to keep in mind, like how do you want to deal with it and what do you want to achieve in the end. And then there are, of course, as for all machine learning devices, a variety of possible input features. You can use the structure by using contact or distance maps. You can even use the 3D structure, so actually use the coordinates. You can use just the sequence. There, of course, you need to find a suitable encoding, so you can just use one hot or something more sophisticated like Elmo. With a sequence encoding, you always, for binding residue prediction, at least have the problem that these input features often get really big. So Elmo, for example, is 1024 values, so you have a 1024 length input feature vector, which is usually pretty long if you have 1000 proteins to train on. So that's something to keep in mind. You can also use predicted features, like for example local structures or secondary structures. Solvent accessibility is often used. Solvent accessibility makes sense because you would assume that the binding site is on the surface and not buried within the protein core, so looking at that can help. And also general amino acid properties, like Yannick already said, so the hydrophobicity, the size, the charge. So I mean, if something positively charged is binding, then you probably need something negatively charged to bind it. And that's, again, important. So that's, you can only use that, or really use that, if you have something that is ligand specific. Because depending on the type of ligand, you might don't need something that is charged, or you don't need something that is hydrophobic. And you can also use sequence variation, and this should be at least widely applicable, because it basically just tells you, okay, if I have a residue and if I mutate that residue and that disrupts the function, then it's probably a functionally important residue, so it could be a binding residue, and that should be independent from what actually binds. Okay, so these are the basics, and now in the last part I want to show you some methods. I mean, you already discussed some also in the exercise. But yeah, so it should just give you an overview of what can be done, the wide variety of inputs, the wide variety of machine learning devices, just to give you an overview. The thing is, so I'm not comparing any performance, because that's really hard, because they use different data sets, they use different definitions of what they define as binding, they sometimes train on very ligand-specific data sets, they sometimes have a broader application. So that's really hard, and that's also what makes it difficult to predict binding residues, and also to claim I'm state of the art, because depending on what you're looking at, it's really hard to define. So let's start with a bit of self-advertisement. So that's a method that I developed. So it's called BindPredict, and I'm currently also working on improving it and developing it further. The first version that we published at some point used basically output from SNP2, so where you basically get the effect of a mutation. Then evolutionary couplings, so where you identify positions that are co-mutating, so if one of them mutates, the other has to mutate, for example, to compensate a structural change. And so that's usually used for structure prediction. What we did is that we identified hotspots, so positions that are correlated with many positions, because they seem to be important for the protein and they could be functional. So that's how we used evolutionary couplings. Then we used solvent accessibility and conservation scores. Solvent accessibility I already discussed, and conservation scores are also rather obvious because a functionally important residue should be conserved through evolution. So as long as the protein still has the same function, the binding site is probably also conserved. And then I use all of these input features as an input to a neural network. It's just a normal shallow ANN with 100 hidden units. It's nothing deep or anything. And then you get a per residue prediction out of it. And here you can see it mapped to the structure. So in fact, I don't use any structural information, but you can, of course, map the predictions back to the structure if you know it. And the thing that I mainly observe, which is also why I show this picture, is then that you have many false positive predictions. So you get many binding residues predicted that are not annotated. And this is an example for that. So here you see like this black thing is the predicted binding site. And what you can observe is that often these predictions look like a binding site, but they are not annotated as one. And that's also one of the major problems when you want to predict binding sites, that you are missing a lot of annotations. So yeah, that basically limits always the performance. But you could also argue it's not a binding site, but that's basically the problem, that you cannot deal with that you don't know if that is actually a wrong prediction or if it's just a missing annotation. Yeah, and as I already said, so I'm still working on that. So currently I have a version where you can remove evolutionary couplings and you still have the same performance. I'm also working on integrating the ligand specific information of these things, but it's ongoing work, that's why it's not included here. But it's, yep.
Yeah, when you have such a situation where you don't have the annotation, or it's it's just a wrong prediction, is it then commonly given to, like, there's probably also you can say that it's probably something that should be annotated. Is it then given to biochemists or something, that they actually look into it, or are they not interested in that?
So I think in general they would be interested.
Yeah.
For us, so at least I don't have the contacts to people like that, so we don't give that out, but that would be the idea, and that's also what we argue in the publication, that we say, okay, the prediction tool is interesting for biologists because they can at least get a hint, this could be a missing annotation, I will investigate that further. And also for these, so these are not like just predictions, but these are the 5 most reliable predictions. So these are the predictions where we're rather sure that they should be correct. And yeah, but that would be, on the long run, that would be the idea. So we don't have that collaboration, but it would be the idea to actually use these predictions to then do further experiments. Okay, so let's move to something completely different, which is ligand RFs. So what they do, they use a sequence profile as an input, which is also something that is commonly done. And then they use 34 amino acid properties that they identified from a list of 500 as the most informative ones. They also consider neighboring residues, so they also have a sliding window, which makes sense because, of course, the environment influences the residue that you are considering right now. And the really interesting thing that they did is this 15 balanced data sets. So with binding residue prediction, you have the problem of that you have a really high class imbalance. Roughly 10 to 9% of the residues in a protein are binding, which is expected because not much of the protein is actually the binding site, but that gives you a really high class imbalance for your prediction task. And what they did with these 15 balanced data sets, so they created 15 data sets where they always took all the positive samples, and then the other half of the data set were randomly sampled negative samples. So you have 15 data sets, always the same positive samples, but different negative samples for all 15 of them. And so they created balanced data sets, and it's kind of a way in between undersampling and oversampling. So they don't lose any information, so they don't undersample because they train on all the negative samples at some point, but they also don't have to generate any artificial samples by oversampling or something like that. And then they use these 15 balanced data sets to train 15 different random forest classifiers. And then in the end, they combine these 15 classifiers to one ensemble classifier. And then they can predict with that. Their prediction is also ligand type specific, so they don't do a general prediction, but they kind of need to know what is binding. Then we have target sec and target com. So here the interesting part is mainly the target com part, but let's start with target sec. So target sec is a sequence-based predictor. So they take the protein sequence, they calculate a PSSM that predict local structures, so secondary structure and solvent accessibility, and they also calculate conservation scores. They take all of that as features, and then they train an SVM, and on top, so they train multiple SVMs, and on top of these SVMs, they put an adder boost to combine all of these SVMs to a final ensemble classifier. And then they can predict binding residues in a protein, only given a protein sequence. And what they then did with target com is something that is also something that we did, for example, with log3, and that also JJ did with his method, that you combine this de novo approach with a template-based approach. So what they do for target com, they take coach, cofactor, TM site, and S site, which are 4 template-based prediction methods. They all work in a very similar way. So coach is basically an ensemble classifier using these 3 and 2 other methods. Cofactor and TM site use structural alignments to identify similar binding pockets. And S site actually does a sequence profile to sequence profile comparison to identify a sequence motif, so they don't need a structure. They still infer annotations from a similar protein, but they don't need a structure for that. But in the end, they all are template-based in some way. And so what target com then does, so you run the predictions for all 4 of them, and if none of them can give you a reliable prediction, so they can't find a template that is similar enough to your protein, then target sec is run, and then you can do the de novo prediction. And that basically combines best of both worlds, because we know that the template-based methods are much better than any de novo method if we have a template. And if we don't have a template, we can still use target sec, which is a pretty good template-free method. So that's the idea behind target com. And last but not least, we have the obligatory deep learning approach that also exists, of course. Here we have DeepSight, which actually uses a 3D CNN. So they went really deep. And they also do it a bit differently. So they don't really focus on actually predicting a binding residue. They can, so that in the end they have for every residue a probability, and they can do a classification in binding and non-binding. But they more focus on identifying the binding pocket on the structure. So they're using structural information, and in the end they map it back to the, or they don't map it back to the structure, but you get the output for the structure, and they validate mainly based on what we predicted, how close is it to the ligand, and does it look like a binding site. So they don't care if there are like 2 or 3 residues that are wrong. They just focus on actually predicting the binding site as a whole. So that's a bit different from the other approaches. And what they do, so as I already said, they use a 3D CNN, so they actually use the structure, they use the 3D coordinates of the structure, and they represent the protein structure with 8 different channels that basically just reflect different propensities of the protein. So they have this hydrophobicity channel. These are for aromatic amino acids, and they have if a hydrogen bond can be formed, so if it's a acceptor or donor. They have the charge, so basically positive and negative charge. They have if any metal ions are in there, and just the size of the amino acids. So it's just 8 different channels. It's just 8 different ways to present a structure, and it kind of follows the idea as always with CNN from image processing, that you also have different channels in an image, and then they just put it into a very deep machine learning architecture, and as I already said, in the end they mainly get binding pockets out of it. They can do a classification, and they also show in their paper just precision and coverage for binding or pair residue prediction, but they focus more on in the structure identifying something that is close to the ligand, that forms a sphere around the ligand that makes sense. So they focus more on the actual binding site instead of the binding residues that are predicted. So yeah, in conclusion, I hope you can see that there's a variety of tools that you can use, and that there's also a variety of approaches that have been tried, and they're all focus on different aspects, and they all cover different things in the end, which makes the field interesting, but also makes the field hard, because there's a lot to consider if you actually want to predict a binding residue, and it's not just so straightforward as maybe other prediction tasks, starting with constructing a reliable data set, as I already showed. So I hope it gave you a good overview. Do you have any questions? Further questions?